The RNA which is both structural as well as catalytic role. That is ribosomal RNA. That is ribosomal RNA. Ribosomal RNA, that is rRNA. Ribosomal RNA, that is rRNA. It is contained in ribosome. This RNA is found in ribosome. And ribosome of eukaryote, prokaryotic cell, 30S type and 50S type, collectively 70S type. These are the two subunits of prokaryotic cell. Two subunits of ribosome in prokaryotic cell. 30S RNA has 16S type of rRNA. Whereas 50S unit has 5S type and 23S type of rRNA. 5S and 23S type of rRNA. Out of which 23S sRNA, that is contained in 50S subunit. Contained in the 50S subunit. This RNA acts as catalyst. Acts as catalyst. 23S type of RNA acts as catalyst, responsible for the establishment of peptide bond. Responsible for the establishment of peptide bond in between two neighboring amino acid. In between two neighboring amino acid. So this RNA acts as enzyme, that is ribozyme. RNA acts as enzyme, that is ribozyme, and this enzyme is totally non-proteinous. Ribozyme is totally non-proteinous enzyme contained in 50S unit of ribosome. In eukaryotic cell, smaller unit of ribosome 40S type, larger is 60S type. And in 40S ribosome, 18S rRNA is present. In this unit, 18S rRNA is present. Whereas in 60S unit, 3 rRNA, 5S, 5.8S type and 28S type rRNA. Out of which 28S type acts as ribosome. 28S type of rRNA acts as ribosome, responsible for the formation of peptide bond. Responsible for the formation of peptide bond at the time of protein synthesis, and that activity is taking place over the ribosome. That activity takes place over ribosome. That is, translation takes place over ribosome. So that RNA which acts as catalyst is only rRNA. mRNA carries information regarding protein synthesis. tRNA, role of tRNA is to transfer amino acid to the site of protein synthesis, whereas rRNA responsible for the proper arrangement of those amino acids which are carried out by the tRNA. So proper arrangement, that is proper polypeptide chain, is synthesized by with the help of rRNA. 34. In eukaryotic cell, RNA polymerase II catalyzes. In eukaryotic cell, there are three types of RNA polymerase enzyme. In eukaryotic cell, there are three types of RNA polymerase enzyme. Number 1, RNA polymerase number 1, found in nucleolus, and responsible for the synthesis of rRNA. RNA polymerase number 2 in nucleoplasm responsible for the synthesis of hnRNA, heterogeneous nuclear RNA, and that hnRNA after processing forms messenger RNA. So direct involvement in the synthesis of hnRNA, heterogeneous nuclear RNA, whereas RNA polymerase number 3 responsible for the synthesis of tRNA, snRNA, 5S type of rRNA, they are all controlled by RNA polymerase number 3, whereas 5.8 type rRNA, 18S type of rRNA, and 28S type, 3 type of rRNA, and this is seen in your NCERT book. Please read carefully. These are the different types of rRNA in case of eukaryotic cell. So my answer is hnRNA, heterogeneous nuclear RNA. hnRNA responsible for it is synthesized by the DNA with the help of RNA polymerase 2 enzyme. This is done by RNA polymerase 3, RNA polymerase number 3. Similarly, this is also the activity of 3. tRNA is activity of 3. In prokaryotic cell, RNA polymerase enzyme is there, no type. RNA polymerase enzyme is there and ability to synthesize all types of rRNA from DNA, that is mRNA, tRNA and rRNA. This is for prokaryotic cell. It has only one type of RNA polymerase enzyme. We can't say it RNA polymerase 1. It is a simple RNA polymerase and ability to synthesize all types of rRNA. Role of snRNA-P. C. In eukaryotic cell, split genes are found. What is a split gene? The gene in which information is segmented. The gene in which information is segmented is known as a split gene. Informative segments are called axon. And non-informative segments are called intron. These are alternately present. That is axon, intron, axon. They are arranged in this manner over the eukaryotic DNA gene. That is gene of eukaryotic cell, split gene. Gene in eukaryotic cell. They are split gene. What type of gene? Split gene. Splitted means it is only with regards to the information only. That is, information regarding protein synthesis is segmented. Informative segments are called axon, question pre-medical. Non-informative segments are called intron. They are alternately arranged. So such gene in which information is segmented is known as split gene and found in eukaryotic cell. At the time of replication, this entire DNA is transcribed as RNA. this entire DNA is transcribed as a whole, so that in RNA, intron exons are also there. That is, formed RNA has intron as well as an exon. This is exonic portion, this is intronic portion, and again exon, again intron, again exon. But, this DNA is transcribed as a whole, and the RNA formed is also with exon and intron. This RNA is known as hnRNA, heterogeneous nuclear RNA. This RNA is known as hnRNA or heterogeneous nuclear RNA or pre-mRNA. We can say pre-mRNA. That is precursor of mRNA. Precursor of mRNA. Now, one more activity is there. Processing of hnRNA is now must from this particular hnRNA. Processing, that is removal of intron. Processing means removal of intron. These are exon, intron, intron, then exon. They are alternately arranged. This is synthesized by this one. Now, processing means removal of intron. So during the processing, these introns are removed. And for their removal, spliceosome is required. Spliceosome are required for the removal of intron. These introns are removed by spliceosome. Introns are removed by spliceosome, and spliceosome consists of snRNA, that is a small nuclear RNA plus proteins. Small nuclear RNA plus protein. Spliceosome consists of snRNAP. So snRNAP removes intron and connect exons. When introns are removed, the newly formed mRNA or processed RNA is now mRNA. It has exons only. Because introns are removed. Introns are removed by which one? snRNAP. And at one end, capping is done. At the other end, tailing is done. This is now processed RNA, or we can say mRNA. Processed RNA or messenger RNA. This is applicable for eukaryotic cell only. So answer of this question is, that is, addition of adenylate, no. Removes intron from hnRNA, this is absolutely right. That is, removes intron. This process is called processing. This process is known as processing or splicing. Exon and introns, I have already mentioned, that is, they are the part of split gene. Split genes were discovered or reported by Richard J. Roberts. And Philip Sharp. Discoverer of split genes are also called interrupted gene. Both are the same thing. Split gene, they are called interrupted gene also. That's why answer is 4. That is more than 1. Whereas transposons are also called jumping gene. These are called jumping genes. Transposons are jumping genes. They are able to change their position. Jumping genes. Reported by Barbara McClintock. Barbara McClintock. So answer for 36 question is D, because a split gene and interrupted gene are the same thing. A split gene and interrupted gene are the same thing. So correct answer for question number 4 is more than one option, because these two are the same. Transcription unit I have already mentioned, that is made up of 3 parts. Transcription unit is made up of 3 parts. This is DNA molecule and its polarity 5335. This is 53 and this is 35, and joining by means of hydrogen bond. And with regards to the coding strand, promoter area is present towards 5 end and terminator is present towards 3 OH end. Terminator is present towards 3 OH end. This is 5 end, this is 3 end, and in between them structural gene. What is present? Structural gene. This is a structural, this is promoter, this is terminator. So this is transcription unit, consists of 3 parts, that is promoter, structural and terminator. Promoter, binding site for RNA polymerase. Structural gene, actual gene responsible for the synthesis of messenger RNA. Terminator, responsible for this region, responsible for the termination of transcription. Soluble RNA is other name for the transfer RNA. Soluble RNA is other name for transfer RNA. It is soluble in 1-MnACl. Soluble in 1-MnACl solution. So it is called soluble RNA. It is also called adapter RNA, because adapter for amino acid. Adapter, it is also known as adapter molecule also. Adapter. Question 39. This structure, both structures are of transfer RNA. Both structures are given here for the transfer RNA. This is for the transfer RNA. The first one, this is clover leaf-like structure. This is clover leaf model of tRNA. Clover leaf model of transfer RNA. This one. Whereas this one is inverted L-shaped structure. This is 3D structure of RNA. 3D structure of tRNA. 3D structure of tRNA. Inverted L shape given by SH Kim. 3D structure of transfer RNA was given by SH Kim. Who received Nobel Prize for the discovery? I have already mentioned in the past questions, and that is Richard J. Robert and Philip Sharp. These two are the recipient of the Nobel Prize for the reporting of split genes or segmented genes. Operon concept, direct question regarding the regulation of, this is given by the Jacob and Monod. They experimented over Escherichia coli. Regarding the regulation of gene expression. Regulation of gene expression. Operon concept was given by them. Lac operon. There are 2 operons, lac operon and that is second one is tryptophan operon. Lac for lactose, concerned with lactose metabolism. Lac for lactose metabolism. Lac for lactose metabolism. If Escherichia coli, a bacterium, is provided with lactose, then only cell is able to synthesize beta galactosidase enzyme. The cell of Escherichia coli is able to synthesize beta galactosidase enzyme. If lactose is there, that is, enzyme synthesis depends on substrate. This substrate is called inducer. This substrate is known as inducer. Enzyme is known as inducible enzyme. And inducer, the system or genetic system which is involved in this particular activity is known as inducible system. So, lac describes inducible system. System was initially switched off by the addition of lactose. System is switched on. This is lac-operon. Consists of set of genes, that is, here I is as present as regulator gene, then P is promoter area, O for operator, binding site of repressor, then 3 structural genes are there, termed as ZYA. These are the 3 structurals ZYA. And this is operator, it is a binding site for repressor, promoter, binding site for RNA polymerase, and I provides repressor protein. I is source of repressor molecule. I is source of repressor molecules which are protein. And binding site of repressor molecule is on operator. Binding site of repressor is on operator. Then only it can regulate the expression of ZYA. In LAC-Z gene, these are the structural gene concerned with the lac-operon. LAC-Z gene responsible for the formation of beta galactosidase. Y gene responsible for beta galactosidase permease. Y responsible for beta galactosidase permease, and A responsible for the transacetylase. These are the 3 structural genes named as ZYA. ZYA. It's a polycistronic gene. Actually. The DNA segment which serve a kind of a switch on and off during, that is operator gene. The answer for this question is operator. See this one. This is lac-operon. And this is the position of operator prior to structural. Structural genes are Z, Y, A. And this is operator gene. And see this one. If on operator, repressor molecules are there. If repressors are on operator, system is switched off. That is, operator, if it is provided with repressor, does not allow RNA polymerase enzyme to cross operator. So transcription does not take place. If transcription is not taking place, then system is switched off. If repressors are removed, then only, from the operator, then only there is a possibility of a transcription. In that case, system is switched on. To switch off, switch on, it is done by the operator with the help of repressor. Repressors are provided by regulator. What is the nature of repressor? Then protein. 45. Question 45. Genomics study along with the human genome. Answer is 4. In all the cases, the complete gene mapping was done. That is, all these are mapped along with the human genome. Human Genome Project, that is complete study of human genome, and it is also with the, that is, these are also already studied in detail. The commonly used host vector for cloning of DNA. DNA cloning, that is, if DNA is added in that area, its copies are made. And it is, in case of eukaryotic cell, it is YAC. This is best. YAC for eukaryotic, for prokaryotic BAC. YAC, yeast artificial chromosome. This is a part of HGP, Human Genome Project. In HGP, DNA fragments were sequenced and by automatic. That is informative question. That automated sequencer was used by the Frederick Sanger. Frederick Sanger has used. DNA fingerprinting recognizes the difference in both A and C, satellite DNA and repetitive DNA. Satellite DNA, that is, certain short sequences are seen number of times. VNTR, that is variable number tandem repeats. Variable number tandem repeats. Variable number tandem repeats. That is known as VNTR. These are the satellite DNA. Satellite DNA, they are present number of times in a DNA molecule. Technique of DNA fingerprinting. In this technique, southern hybridization is used. Southern hybridization, that is, DNA is extracted from the source and by the process of gel phoresis, it is arranged in sequence according to their size. By the gel phoresis process, they are arranged according to size in agar gel. Then after that, they are shifted on a nylon membrane in single stranded state. Shifting of DNA on nylon membrane in single stranded state. This is southern blotting. Northern blotting applicable for RNA, western blotting for protein. Northern blotting is applied in case of RNA, whereas western blotting is used for the proteins. Western blotting is useful for the proteins. That's all for the DNA. Thank you.